Hi my dear students, welcome to my biology class. Today, let's begin our class with a latest news on coronavirus. You just watch it. Mutation allows coronavirus to infect more cells. Study finds. Coronavirus mutation makes it even more infectious and is emerging as the dominant kind, research finds. Mutation could make coronavirus more infectious, study suggests. Did you watch the news on coronavirus and COVID-19? We all are familiar with the coronavirus and COVID-19 as we have been experiencing a pandemic situation globally in the context of COVID-19. You might have heard about mutated coronavirus, how these viruses become mutated. So let's study today about mutation. So today's topic is mutation. So after studying about mutation and its facts, you will come to know what is the meaning of actually the mutated coronavirus. So let's start now. Our topic is mutation. So mutations are random, unpredictable changes that occur in the genetic material of an organism that results in the changes of the genotype and the phenotype of an organism. So what are the 2 points in the mutation? First of all, it is the alteration of the genetic material. Alteration of the genetic material The genetic material could be RNA or DNA. What is the second point? It results in the changes of the genotype and the phenotype of an organism. So, results in changes of the genotype and the phenotype. Phenotype of an organism. It means that if there is a change in the gene, the information that is used for controlling a trait in the organism, then what would be the result of the genotype? The genotype will be altered. So ultimately, that results in the change of the phenotype. So finally, a different phenotype is expressed in the organism. The term mutation was coined by Hugo de Vries in 1901. Hugo de Vries in 1901. He observed a number of mutations in the experimental material evening primrose. What was his experimental material? Evening primrose. Its scientific name is Enothera lamarckiana. Enothera lamarckiana. The mutations are occurred due to the action of certain factors. Those factors are known as mutagens. Mutagens. So, mutagens are the mutation generating factors. Those factors are categorized as physical factors and chemical factors. Physical factors and chemical factors. The physical factors like ionizing radiations and non-ionizing radiations. Ionizing radiations and non-ionizing radiations. The ionizing radiations are alpha rays, beta rays, gamma rays and X-rays. The non-ionizing radiations are UV rays, ultraviolet radiations. UV rays. Okay, the chemical factors are certain kind of chemicals like base analogues. Example, base analogues, alkylating agents, even nicotine can act as a mutagen. So, nicotine, acridine dyes, These are some examples of chemical factors that can generate mutations in some living organisms. Mutations can be occurred both in chromosomes and genes. So, it is categorized as chromosomal mutations and gene mutations. Chromosomal mutations and gene mutation. Chromosomal mutation means the mutations occur in the chromosomes. Gene mutation means the mutations occur in the gene. Then let's come to the point, chromosomal mutations. Chromosomal mutations are of two types. They are morphological or structural chromosomal mutation and numerical mutation. Structural or morphological. And the next category, numerical. Numerical chromosomal mutation. Okay, structural morphological mutation means the structure of the chromosome can be altered by certain kind of factors. There are four types of structural chromosomal mutations. They are deletion, second one duplication, third one inversion, fourth one translocation. Numerical chromosomal mutation means the number of the chromosome can be changed by certain kind of factors. So, the numerical mutations are of two types, euploidy and aneuploidy. It is divided into two categories, euploidy and aneuploidy. Okay, then gene mutation. Mutation that occurs in the gene. It is of two kinds. They are point mutation and frame shift mutation. Point mutation, frame shift mutation. Okay, this is the classification of the mutation, chromosomal and the gene mutations. Now, let's study some basic facts on the chromosomal mutations and the gene mutation. First of all, chromosomal mutations. Chromosomal mutations. The first one, deletion. In this, a segment of the chromosome is get deleted. Okay, a segment is deleted or lost. So, for example, suppose this is a chromosome with some genes A, B, C, D, E, F, G. Okay, so suppose if this particular part is deleted from this chromosome, the structure of the chromosome will be changed. So, the structure look like this, A, B, C, D, E. So, F and G has separated from this chromosome so that the structure of the chromosome changes like this. This is deletion. The second one is duplication. Duplication. It is the duplication of certain segment of the chromosome. Duplication of a segment. So for this also we need to draw one chromosome A, B, C, D, E, F, G. Suppose if this particular segment is duplicated D, E, F, the structure of this particular chromosome will be like this: A, B, C in one arm. In the next arm will be having duplicated D, E, F, again D, E, F, then G. Okay, this part is duplicated. Got it? The third one is inversion. Inversion. It is the reversing of a particular segment of the chromosome. Reversing of a segment. Suppose this is a chromosome with the genes. If this particular part DEF is reversed, so the chromosome look like this. See, the structure will be changed like this: A, B, C. So F, E, D, F, E, T. Okay, then G. See the reversing of the particular segment. So it causes the reversing of the genes in that area. The fourth one is translocation. Fourth one, translocation. Translocation is the exchange of segments between the non-homologous chromosomes. So for this we need to consider two chromosomes, non-homologous chromosome. One chromosome is with the A, B, C, D, E, F, G genes. Another chromosome with the 1, 2, 3, 4, 5, 6, 7. So these two are non-homologous chromosomes. Non-homologous. So here a segment is separated from the one chromosome and is attached to the next non-homologous chromosome. So if this particular segment is separated from this chromosome, it will be attached to this. Then at the same time, if it is separated from this chromosome, it will be attached over here. So it can be represented as A, B, C, D, then 5, 6, 7. Then the next chromosome 1, 2, 3, 4, E, F, G. So as it is exchanged in between the non-homologous chromosome, this type of translocation is also known as reciprocal translocation. Reciprocal translocation. Did you get it? These are the 4 types of structural or morphological chromosomal mutations. The next one is numerical chromosome mutations. Numerical chromosome mutation. Chromosome mutation. The 2 types are euploidy and aneuploidy. Euploidy and aneuploidy. Euploidy occurs due to the failure of cytokinesis during cell division. Cell division consists of 4 stages: prophase, metaphase, anaphase and telophase. After these 4 phases, cytokinesis must happen. So look at here. See, suppose this is a cell, and here at the end of the telophase stage, the cell look like this. That means the opposite part of the cell will have equal amount of genetic material. So ultimately the cytokinesis, the division of cytoplasm must happen here. Then only, this is cytokinesis, then only it would divide into two equal or two similar cells with a similar genetic makeup. If it doesn't happen, what is the result? So the doubling of the whole set of chromosome in the cell. So in the resultant cell. So instead of these two, the one cell form with the two sets of chromosome. So instead of a haploid, it has one more set of the whole number of chromosomes. That is euploidy. So euploidy occurs due to the failure of cytokinesis. So what's the reason? Failure of cytokinesis. Okay, then you know that n number of chromosome means haploid or monoploid. Chromosome means haploid. 2n means diploid. 3n means triploid. 4n means tetraploid. 3n triploid. 4n tetraploid. Here, if it consists of more than two sets of chromosome, this condition is called polyploidy. Polyploidy. 3n, 4n, 5n, 6n conditions are polyploidy conditions. These polyploidy conditions can be seen in plants. Example, plants. So did you get it? The reason of euploidy. The next is aneuploidy. It is occurred due to the addition or deletion of one or two chromosomes to the total number of diploid set of chromosomes. So it can be represented as 2n plus 1, 2n minus 1. Okay, then 2n plus 1 means addition of a single chromosome to the 2n set of the chromosome. Suppose this is one pair of chromosome, addition of one more chromosome. That is 2n plus 1. This condition is called trisomy. So 2n minus 1 means, suppose this is a pair of chromosome. If one is absent, this is called monosomy. Okay, so the result will be a single chromosome, monosomy, that is 2n minus 1. The next one, when two chromosomes are added to the 2n set of chromosome, 2n plus 2 is equal to tetrasomy. Okay, one pair plus again 2. So that is tetrasomy. 2n minus 2 is equal to nullisomy. 2n minus 2, that is nullisomy. Here, from the original diploid set of chromosome, two chromosomes are deleted or absent. So such condition is called nullisomy. So 2n plus 1 is trisomy, 2n minus 1 is monosomy, then 2n plus 2 is tetrasomy, 2n minus 2 is nullisomy. These are the different types of aneuploidy condition: monosomy, trisomy, tetrasomy and nullisomy. So can you guess the reason of aneuploidy? The reason is the failure of segregation of chromatids during cell division. So when chromatids fail to segregate at the time of cell division, they try to stay together so that it causes the addition of one chromosome. Sometimes it leads to the deletion of one chromosome, one or two chromosomes. So that is the reason of aneuploidy. The aneuploidy conditions lead to different types of abnormalities in living organisms, including human beings. Next, let's study about gene mutations. Gene mutations. So, what are the two types of gene mutations? Point mutation and a frame shift. Point mutation and a frame shift mutation. Point mutation means if mutation occurs at a single base pair nucleotide of the DNA, that results in an abnormality in the gene that ultimately produce a false protein in the body of organism. That is called the point mutation. So for this, to understand more about this, we need to consider the segment of a DNA. Suppose this is the segment of the DNA. The DNA is made up of nitrogen bases, sugar and phosphate. So when we draw the segment, we need to consider the base pairs. So here I draw one hypothetical segment of DNA with the base pairs adenine, thymine, thymine, adenine, guanine, cytosine, cytosine, guanine, guanine, cytosine. So this is a hypothetical sequence of DNA. Then this is a template strand with the 3' to 5' polarity. Another coding strand with the 5' to 3' polarity. Suppose this particular segment of the DNA consists of the information for synthesizing a particular protein. That particular protein controls a trait in an organism. Look at here, in the production of protein synthesis, only a particular part of or particular strand of the DNA is involved, that is the template strand. So this particular strand is involved in the protein synthesis. So here I draw ITGCG template strand. If there is any change in the single nucleotide, suppose if thymine is changed as guanine, and the sequence of the template will be because of mutation. If it is changed by mutations or mutation, so the sequence will be arranged like this A, G, G, C, G. Okay, then so the complementary strand will be thymine, cytosine, cytosine, guanine, cytosine. See how this DNA segment is different from the first one. Okay, this is called point mutation. A change in the single nucleotide base causes the alteration of the DNA segment. Thereby, an abnormal protein will be synthesized. So, abnormal protein, then finally an abnormal trait. Sickle-cell anemia is a classic example of the point mutation. Sickle-cell anemia. Example, sickle-cell anemia. Then did you get what is point mutation? It's a single base pair change that results in the alteration in the DNA, so thereby the alteration in the trait, that is point mutation. So here I can write single base pair change in the case of point mutation. Then what is frame shift mutation? If more than one nucleotide base pair is changed, suppose a long sequence of the DNA like this, if a particular segment is changed or altered by mutation, that would result in the same case abnormal protein and the abnormal trait in the organism. That is called frame shift mutation, deletion of a particular segment of DNA by mutagens. Deletion of a segment of DNA causes the frame shift mutation. Example, cystic fibrosis. Example, cystic fibrosis. Okay, here we have learned only the basic facts on the gene mutation. What is the actual cause of this gene mutation? Here the DNA is the genetic material. The DNA, if DNA is altered, if any part of the DNA is altered, that would result in the abnormal production of particular proteins, and thereby finally it affects the phenotype of the organism. That is called gene mutation. Now, did you get the type of mutations? The chromosomal mutations and the gene mutations. Then, let's come to the point that we have discussed in the beginning about a mutated coronavirus. That, as we all know, that coronaviruses belong to a group of RNA viruses. That means the genetic material of coronavirus is RNA. RNA is a polynucleotide, a single-stranded polynucleotide. It is highly reactive as it has an OH group in the ribose sugar. So, as it is highly reactive, it undergoes mutation very fast. That may be one of the reasons of the formation of mutated coronavirus. So, did you get what is the actual meaning of mutation? Alteration of the genetic makeup of an individual. I hope you understood the topic well. With this, I conclude my session. Thank you and have nice moments.